fertilization is fusion of male and female gametes and it forms zygote. So this chapter, sexual reproduction in flowering plant, it also includes male and female gamete fusion. In angiosperm, there is a specific feature that angiosperms have double fertilization. So two male gametes are going to produce and they are going to fuse with egg cell of the female reproductive system of the flowering plant and the primary endospermic nucleus. So here double fertilization is taking place. दो male gametes है, एक egg cell है वहाँ पर, female reproductive system एक egg cell है and उसके अलावा primary endospermic nuclei है, दो fusion है, hence it is a double fertilization. And also there is a triple fusion. A nucleus that is going to fuse inside forming a one cell, that is endosperm, is actually triploid. It is having three nucleus by fusion. We'll see how this unique feature occurs in angiosperm, but before that, how the gametes are formed. So first of all, we are actually checking out that how male gametes in flowering plants form. So main reproductive system in flowering plant is anther and filament that are a part of the reproductive system, that is stamen. So stamen is a male reproductive system, or we call as stamen is an endoecium. This endoecium carries anther and filament. Filament is just to attach the anther to the flower. So main thing is anther. So stamen is also known as microsporophyll. Simultaneously, we also can say that anther is microsporangium. Attend to the meaning why I'm saying this microsporangium, microsporophyll, but these are the terms according to their development, according to the structure. So let's see the anther first of all, this microsporangium, where the pollen grain, future pollen grain are going to form. This anther, when we found, this anther is a bi-lobed structure. This anther is a bi-lobed structure. So we can see this, it is having lobe 1 and lobe 2. Right? Now, when it cannot be seen on the microscope while before cutting, so it should be cut from here. So with the help of blade, it has been cut. Now when this cut has been made, जब हम इसमें cut बनाएंगे, तो हमें क्या देखिएगा? इस तरह से एक लोग यहाँ आएगा और एक लोग यहाँ. But so हमें कैसा स्ट्रक्चर मिलना चाहिए? कैसे? और एक ऐसे और दो होगा इसमें एक फिलामेंट में, राइट? इस तरह से हमें स्ट्रक्चर मिल जाएगा जो कि फिलामेंट में है. बट वी कैन सी देयर इज अ स्लिट हियर आल्सो. यहाँ भी आपको एक स्लिट जो है दिख रहा है. सो इट इज नॉट लाइक दिस बट इट इज लाइक दिस. ओके, सो इट इज लाइक दिस. So this is the structure of anther when we dissect it, when we cut it. From the microscope, when we see a large view of this part, what we will absorb, we'll see is right. So an anther is a bi-lobed structure. Number one, number two. When we enlarge this, we can find that it is having two lobes, and by downward, if we see, it is attached to a filament. Right? So lobe one, lobe two. These lobes are connected by some sterile cells which are not going to produce any kind of pollen grain, that is connected. they are having, each lobe is having two such cell layers, two such thicka. So anther is a bilobe and diaphytous structure. In overall, if we see here, it is having 1, 2, 3, 4 cells, 4 such cell layers. These cell layers are known as sporogenous tissues, which are going to be the future pollen grain. So these sporogenous tissues are, we are having 4 such cell layers which are going to produce the pollen grain. So these sporogenous tissues signifies the stain, because 4 है यहाँ पर हमारे पास 4 ऐसे sporogenous tissue के layer है, तो इसलिए anther tetra in 4 sporangiate structure भी कहलाता है. These are sporogenous tissues. Now these sporogenous tissues, we are having 4 such layers. These sporogenous tissues are going to produce pollen grain or microspores. Pollen grain को हम microspores भी कहते हैं. Sporogenous tissues are diploid and pollen grains that are produced are haploid. So each cell present in this sporogenous tissues is diploid, and each one of them is undergoing meiosis. So each sporogenous cell is undergoing meiosis, and they are going to produce the pollen grain. Each cell is going to produce the pollen grain. Now each cell is going to produce how many pollen grain? So each sporogenous cell is going to be in the meiosis process. एक parent cell से meiosis में कितने cells बनते हैं, कितने daughter cells? 4. तो एक sporogenous से कितने cells बनेंगे? 4. तो 4 pollen grain जो है, 4 pollen grain जो है बन सकते हैं इससे एक sporogenous tissue से. These cells after division used to remain fused together. कभी कभी वो pollen grain के फॉर्म में sporogenous cells जो meiosis करके बनते हैं वो टूटते नहीं है, एक साथ रह जाते हैं. ये जब एक साथ रह जाते हैं इसे हम कहते हैं microsporotetrad. So that's happened inside this structure. I've just told you, I haven't told you the structure, but now we have only discussed what happens inside the structure. So stamen is microsporophyll, anther is microsporangium, which we are sporogenous tissues. Sporogenous tissues undergo meiosis to produce 4 daughter cells which are haploid. These haploid cells are known as microspores. When they remain together, glued in the form, sometimes they remain glued, they are known as microsporotetra. When they divide, they form pollen grain. There is a reason that why they are not dividing, because inside the pollen grain is also there is some stages of division taking place. Something is happening. This whole story is about microsporogenesis. Now what is this microsporogenesis? Genesis is something synthesizing. Spore means we are synthesizing microspore. जब microspore यानी pollen grain रेंडम में बन जाएंगे तो इसको microsporogenesis कहेंगे. It is all from the diploid cell. But male gametes are not formed. We will see it in the next session. Before that, what is the structure of this egg? Of course, we have to read. What is all inside this structure? Sporogenous tissues के अलावा और क्या-क्या layers यहाँ पर हैं? So let's see these layers here. The outer layer, the outer protective layer that is continuous overall. Okay, that will be continuous overall is the epidermis. matures inside cells, pollen grains are formed. So all the water inside the anther can comes down. तो जितना भी पानी का लेवल है वो कम हो जाता है, वो अपनी मॉइस्चर जो है एंथर लूज कर देता है. जैसे प्लांट का कोई भी पार्ट मॉइस्चर लूज कर देगा, वो क्या हो जाएगा, डिहाइड्स टूटने लग जाएगा. एंड दैट इज विद द हेल्प ऑफ अ लेयर कॉल्ड एज endothecium. An important layer, endothecium. This endothecium layer helps in the hyacinth of anther. Now, after endothecium, there is a layer. After endothecium, there is a layer that help pollen grain inside to grow and not to contact with the endothecium layer, so that it will not dehyde. इससे पहले dehyde नहीं होंगे. ये गार्ड करने के लिए there are two layers, middle cell layer. यानी pollen grain जब तक mature ना हो जाए उससे पहले endothecium layer के contact में आएंगे. तो उसके लिए यहाँ पर दो middle layers होती हैं, एक और दो. These are middle layers. Now, after middle layer, there is an innermost layer which is continuously surrounding the sporogenous tissues. Sporogenous tissues को भी surround करती होगी चारों ओर से. पूरे anther को नहीं, सिर्फ sporogenous tissues को. This is the tapetal layer. Now, this tapetal layer has a function that it provides nourishment to the developing microspore or pollen grain. It adds on some of the chemical on the pollen grain to make it distinctive. वो ऐड करती है युविश बॉडी pollen kit so that pollen can be recognized from anywhere. किसी भी प्लांट का आपको पता चल जाएगा उसके ऊपर की थिकनिंग देगी. सो एक ऑर्नामेंटल फीचर जो है वो टपेटल देती है. नरिशमेंट भी वही प्रोवाइड करती है. दैट इज इट्स फंक्शन. That is its function. So number one layer of anther is epidermis. Number two is endothecium that help in dehyde since it have fibers. Number third is two middle layers. And number fourth is the petal. And of course, we should not forget the innermost layer, that is sporogenous cells, sporogenous tissues or cells. These layers are connected with the help of connective. Okay, so I'm just drawing the part which we will read, and then you can see the clear picture of the diagram. So, stamen and anther. Stamen is a microsporophyll. Stamen is a microsporophyll. This microsporophyll is a container which contains a sterile part that is filament, and anther which is a fertile part. Anther is bilobe. Why I am saying it is bilobe? Because it is having two lobes. Each lobe has two sporogenous cell layers. Hence it is diphthous. Then overall anther is having 1, 2, 3, 4 sporogenous cell layer. 4 means tetrasporangium. Extra features are just cutting it out. मैंने पहले आपको बता दिया तो extra features बता रही. जो ये आप स्ट्रक्चर देखते हो कब देख पाते हो, जब आप इसको कट कर देते हो, स्प्रेड कर देते हो anther के अंदर और उसमें आपको ऐसा स्ट्रक्चर दिखता है. वो बटरफ्लाई जैसे क्यों दिख रहा है, ऐसे एक और दो पार्ट इसलिए नहीं दिख रहे बिकॉज एक लोब में बीच में स्लिट है, सेल लेयर्स के बीच में भी स्लिट है, इस वजह से वो इस तरह से दिख रहा है. बीच में एक लेयर है कनेक्टिव जो कि स्टेराइल सेल्स है. आप देख सकते हो सबसे पहली जो लेयर है anther के अंदर है वो एपिडर्मिस. सेकंड endothecium which is having fiber for dehyde cells. Third, middle layers which are protecting or becoming a guard between microspore cells and endothecium till they develop. Then after middle cell there is a continuous layer tapetum which is going to provide the nutrition to the developing microspores, pollen grains. And inside this what is happening? Inside this sporogenous cells are going to divide. These sporogenous cells when divide, they are going to divide meiotically. So first division जो हुई यहाँ पर for the formation of gametes, first division is meiosis. In the term of going to be the gametes, सबसे पहले जो division हुई वो meiosis. Diploid से बने haploid cell and haploid cell में pollen grains. And we can say simply not pollen grains. Initially हम इसे pollen नहीं बोलेंगे, microspores बोल सकते हैं. So here we can see that pollen grain is formed after by this process what we have seen in the later one. Stamen is a microsporophyll which forms microsporangium, which is having microsporangium anther, in which we are having sporogenous tissues which developed due to many cell layers present in the microsporangium structure. And sporogenous tissues, these sporogenous tissues are diploid. जैसे है diploid है, इनके पास two set copy है chromosomes में. Now, two है ये diploid है ये, इनमें meiosis होती है, हर सेल में meiosis होती है, हर सेल, हर parent cell एक से चार microspore बनते हैं. Now they initially are tetrad. In a tetrad form, all the four cells are glued together, and then next is when they just come out, they form pollen grain or the microspore. Let's see the structure of this pollen. Now we are seeing the microgametogenesis. Here we are seeing the microgametogenesis. Now this is started from pollen grain, or we can say microspore, that is a haploid cell. Now in that haploid cell, when it matures, it has two layers, XII and anther. XII is an outer layer made up of a chemical which is a very resistant material that cannot be broken down, and for years it can survive. It can also used as fossil to study the pollen grain of a plant that may occur at the time and in locality. Then also this sporopollen. There is one more fact. The sporopollen is the chemical which is made up of the layer added by the tapetum. Tapetum जो nourishment देता है pollen grain को, वो sporopollen भी बना रहा होता है उस समय. Sporopollen की layer से बना होता है बाहर का XII. When we see under the microscope, what we found there is some ornamental thickening of the sporopollen. कुछ ornamental thickening जैसा structure जो है बने होते हैं, and it is different for all the pollen grains. Sometimes you might see the shape of the pollen grain is like this inside the microscope. Sometimes this thickening is hexagonal. So this thickening over the pollen grain also helps in distinguishing which plant's pollen grain it is, and this is due to the sporopollen. Then there is another layer in time. In time is a layer which is flexible and it is made up of pectin and cellulose. Now that sporopollen layer, that XII layer is very thick, but if the pollen content have to come out, because we know that after the pollen grain male gametes are going to form. Male gametes बनने वाले हैं pollen grain के बाद, तो वो स्टिग्मा के surface पे किस तरह से बाहर निकलेंगे अगर बाहर इतना thick coat है, इतना resistant coat है तो. So there should be some open pores, there should be some open area from that content can come out. So sporopollen is very hard layer, very resistant layer, it cannot be broken down by any chemical, but it have some pores. So those pores are called as germ pore. It have pore, it has pore, so that those pores are called as germ pore, from where the pollen tube will grow, and we can say it as pollen germination because pollen tube is coming out from it. Then you can see there is an intine, a pectin and cellulosic layer. It is comparatively flexible, it is comparatively thin layer and not as hard as sporopollen, and it cannot survive as for generations. This pollen grain is also responsible for many pollen allergies. So those airborne diseases and also some pollen-borne allergy are in the same case, because these pollen grains are having a very dry. Sometimes they are very dry and they can taken out by the wind. And in some trees like the Parthenium grass that have some pollen grains which can cause some pollen allergies. This pollen is filled with many sugary content that is in somehow used with some medicines also. So this pollen grain is a very point of structure, an important point of structure. Then what happens inside this pollen grain, let's see. So first division has taken place here. First division is meiosis. In the direction of formation of male gamete, first division is meiosis. Now all the cells are now haploid when we are talking about pollen grain and afterwards. All the cells are haploid. So meiosis definition, first division is done. Now second division takes place in the direction of forming male gamete. Second division is mitosis. Now, inside the pollen grain, there is an unequal division, mitosis, which divides it unequally into two cells, vegetative and generative. Two cells are formed. Now, what happens? Initially, it is a whole vegetative cell, and inside it, there is an unequal division of the nucleus. So, in a large vegetative cell, there is a vegetative nuclei and a small generative nuclei. Wall formation is not followed by the division at just a time. एकदम से डिवीजन नहीं होती और एकदम से डिवीजन हो जाने के बाद सेल वॉल नहीं बनती. तो वेजिटेटिव सेल होता है, वेजिटेटिव न्यूक्लियाई होता है और जनरेटिव न्यूक्लियाई होता है. After some time there is a wall formation, so there will be a larger cell and a smaller cell in a pollen grain. After second mitotic division, after some time, vegetative cell and smaller generative cell. Now there will be now subsequently in a synchronization there will be third division that is again mitosis, because already the chromosomal number become half, right? It become one. Now it cannot be half. So two generally diploid plants, so two, then become one chromosomal copy, and now it cannot be half. So the division will be only mitosis to increase the number of the cell. Now vegetative cell is there. Then there is generative cell. Now second type of mitotic division और overall अगर हम बात करें third division जो होगी वो किसमें होती है? Generative cell में. This generative cell divide and form two male gametes. Now what is the function of vegetative cell here? It has helped to form the pollen tube. During the pollen germination, when it is throwing out its content into the style, okay, when it reaches to the compatible stigma, it is releasing its content via pollen tube. So it have to come out, it have to release its content, it have to release the male gametes. So for that, vegetative cell helps to form the pollen grain. And generative cell, that means it is for the next generation, it is for generating something that will undergo the mitotic third division and forms two male gametes. And this is all about the microgametogenesis, and before that microsporogenesis, and how the male gamete forms in flowering plants.